Hi, my name is Sohail Antoine Younis. I'm an assistant professor at Emory University in the Pathology Advanced Translational Research Unit, PATRU. We study how the gut bacterial flora modulates immune cells in HIV infection. Our findings indicate that gut bacterial toxins impair CD4 T cell restoration during treatment by anti-HIV drug. Uh, Specifically, we study a category of HIV-infected patients called immune non-responders to therapy. These patients maintain low CD4 T cell count even after therapy, and this is linked to morbid and mortality and morbidity. In 2018, in a manuscript published in JCI, we have shown that CD4 T cells in INR have mitochondrial defect. Mitochondrial network is essential for cell function, and cells are unable to survive with dysfunctional mitochondria. Here we show an electron microscopy section of CD4 T cells from healthy donor compared to CD4 T cells from ANR patient. This is the nucleus here shown, and these small organelles are healthy mitochondria, dense, well-shaped. These healthy mitochondria are rarely detected in the section from INR, and this is linked to cell death. So what are the drivers of this mitochondrial defect in CD4 T cells? One approach that we used is to analyze the metabolic profile of CD4 T cells from INR and IR and healthy donor, healthy controls. We isolate CD4 T cells from healthy donors as well as from IR and INR, and we quantitated the metabolite. Then we correlated and validated the role of metabolite in mitochondrial function and CD4 T cell count. As shown in this figure, we found an enrichment of picarizole sulfate in INR CD4 T cells, as shown in here. PCS is a picarizole sulfate is a metabolite coming from the gut as a result of bacterial activity by the gut bacterial flora. Using two other cohorts, one shown in green and one shown in red, we found high levels of picarizole sulfate as well as indoxyl sulfate in the plasma. The latter also is also derived from gut bacterial activity. In here, the y-axis shows the concentration of PCS and IS, and the y-axis in the x-axis we show CD4 T cell count. As shown, there's a negative correlation between levels of PCS and IS and CD4 T cell count. And the blue dots here are the data coming from INR patients. We also found that treatment of CD4 T cells from healthy donor with PCS or IS block CD4 T cell expansion and proliferation and render the mitochondria unhealthy. This is not shown here, yet you can find it in the published article. In here, electron microscopy section from healthy donor, IR, INR, and healthy donor treated with PCS are shown. In healthy control and in IR CD4 sections, we detect active dense mitochondria as shown in here. Yet in the INR or the healthy control treated with PCS, round-shaped inactive mitochondria are displayed. And here in the bottom panels, in which the number of mitochondria and the inactive versus active mitochondria are shown, indicating that PCS-induced mitochondrial PCS-induced inactive mitochondria, similar to findings among INR CD4. This is also here an alternative measurement of inactive mitochondria as shown in D. Thus, PCS blocked CD4 cell proliferation and rendered mitochondria in CD4 T cells inactive. We obtained stool samples from healthy donor INR and IR, and we analyzed the bacterial diversity by molecular techniques described in the article. We found that stool samples from INR are enriched in proteolytic bacteria able to produce PCS, such as Bifidobacterium, Rhinococcus, or Lactobacillus. We propose this model in which we say that the enrichment of proteolytic bacteria in the INR, in the gut of INR, would lead to higher production of picrazole sulfate that affects mitochondria in CD4 T cells and lead to CD4 T cell death, as shown in here. So strategies to lowering proteolytic bacteria may be beneficial to reducing gut bacterial toxin levels and restoring CD4 T cell count in INR.